MIT. The whole area around MIT was awful, but now it's, it's just totally transformed.
Exactly. So we'll get into that. I'm gonna, are we ready to roll, everybody?
We are rolling.
Am I okay in terms of on camera for you, Bart?
There's no way with this lens, but with this
All right.
lens you're both.
Oh, okay. Well, we'll move in and out. Okay. Steve Chapple with Intellectual Capital. I'm here with Steve Chapple with Intellectual Capital, NBC. I'm here with Dr. Robert or Bob Langer, one of the finest bioengineers on the planet today. Dr. Langer, you're both a bioengineer and a chemist who developed time drug release and tissue engineering and ways to shrink tumors and much more. You hold over 1,000 patents. It could be said that biotech would not be possible without these contributions, and an incredible 2 billion people benefit from your inventions in some way. But I was quite interested in the nature of scientific creativity, the way you've jumped to different fields. How does a scientist come to be creative?
Well, for me, and I think it's possibly true for many others too. So, I mean, and this is just one way, is like I was a chemical engineer. I got my undergraduate degree in chemical engineering, my graduate degree in chemical engineering, and when I was a postdoc, and I don't want to give myself any credit for this, but I did something totally different. Most of all my colleagues actually went to the oil industry because that's what chemical engineers did at that time in the 70s when I was getting my degree. But I wasn't excited about that, so I ended up doing something totally different. I actually went to work in a hospital in a surgery department, and I was the only engineer there. And I got exposed to so many things that were so different from what I'd learned before. And so I think by knowing the fundamentals of, say, one discipline, chemical engineering, and then getting exposed to a totally different discipline, say surgery and medicine, that just gave me a lot of ideas because I approached surgery and medicine very different from anybody else just because my background was so different. And I think in a broader sense, to answer your question, when I think of some people who are creative, it's because they've been exposed to such different things, and then they put them together in unusual ways. I think there's other aspects, obviously, to creativity than just what I said, but I think for me that was a very important thing.
Okay, and we were talking to Dr. Witten earlier, and his father was a theoretical and is a theoretical physicist. But your dad ran was it a corner liquor store?
He ran a small liquor store. That's right, in Albany, New York.
So that's kind of cool. It's like Bill Clinton almost in the South. But how do you get from that to to these great leaps forward in biochemistry and bioengineering?
Yeah, well, my dad ran a small liquor store, but it's interesting. I mean, he grew up in a very different time. He grew up, actually, he was a very smart guy, probably smarter than me. He was at Union College, and he finished, I think, second in his class, and he went to Harvard and actually got a master's degree in English. But
that's wild.
But he graduated in 1937, and that was the Depression, and I remember he said the only 2 jobs that he could get were like teaching in North Dakota or something like that, and he wasn't that excited about it, and so, he didn't end up doing that. And I think also when you went through the Great Depression, he felt like he was really concerned because he saw so many people go from having some money to nothing, that he wanted to have a secure job where he could provide for his family and make sure that we got good educations. But even then, even when I was a little boy, one of the things that he and my mom and my grandfather did is they got me these sets, these Gilbert chemistry sets, Gilbert Erector sets, and Gilbert microscope sets, and I loved those. Those were great. I would play around, mixing chemicals together. I mean, I didn't know what I was doing, but I really enjoyed it. And I think that was maybe the start of me, being more curious and thinking about science. And he also, would spend a lot of time playing math games with me and things like that. And I think the other thing that was really good about my parents is they said, well, really, both my mom and dad, their major concern was just that I'd be happy. It's like not that they wanted me to get all A's or anything else, just to be happy. And I think that was very helpful in, my early formative years.
Okay. Back on creativity, this is a pretty cool book, actually, written by Kazuo Inamori, who's written a few, and he's, of course, the founder of Kyocera.
Yes.
and a creative scientist or applied scientist in his
Certainly.
own right. So I wanted to read that. The way to true creativity is not through the accumulation of existing scientific knowledge. It starts with a leap of inspiration. This inspiration forms a philosophy, and when it is proved and accepted, it finally becomes science. This is his opinion. True creativity blooms when we dare to reject common sense and traditionally accepted scientific knowledge. Does that apply to some of the things you did, or?
It does, especially the last part. You know, I would say that there's something that, of course, that a term that I often use called conventional wisdom.
Right.
And the point I guess I'd make is conventional wisdom is often wrong, but you're taught it your whole life in class and articles and things like that. And I know that my early career, a good part of what we did in terms of the work we did on polymers and plastics, you know, went against conventional wisdom. And of course, sometimes when you do that, you also pay a price because the scientific community doesn't accept what you do. And I know in my case, it had a big effect on my not getting research grants. In fact, even almost not keeping my faculty appointment at MIT. But I think that's often the case, that to be creative, somehow you have to go beyond what conventional wisdom is. And I think so. I think what he's saying probably applies to many people. And I think what I said doesn't apply just to me. It applies to many people in many fields.
So it's a bit more cutthroat there? I mean, given the great advances, back then it would have been drug delivery, right? Crossing different fields. That was seen as unorthodox?
Well, I think there were two things, you know, at least two things that were maybe unorthodox. One is just the field in general. I was a chemical engineer, and I remember when I went from my chemical engineering background to now working in a hospital and a medical school, when I applied for faculty jobs, no chemical engineering department would hire me because they just said what I was doing and what I was proposing wasn't chemical engineering. So that was one aspect. I think another aspect was this more specific work itself. We made a discovery, which we published in the journal Nature, that we discovered certain ways that you could modify plastics so that we could use them to release slowly molecules of any size. And that did go against the conventional wisdom in the scientific community for a long time. And so a lot of scientists, when I wrote about that and talked about that, thought it had to be wrong, and so they rejected the grants, and they probably didn't say such nice things about me.
But it proved out in many other labs. What was
later on, yes, later on, but that takes time. You know, in other words, whenever you make findings, you know, sometimes there's a significant period of kind of rejection and skepticism. I think Dr. Folkman once told me that.
And he was your mentor?
He was my mentor, yes. He went, and he made some great discoveries, and he once said to me, it was something like this. He said, first, when you, you know, first you discover something and people say, you know, it can't be right, and then, you know, you go further and they, you know, they keep criticizing it, but then many years later they say, well, okay, we did it, and take credit for it, so to speak. You know, and so I think that you go through different stages like that.
Well, you know, to an intelligent lay audience, what is a polymer? And when you say a molecule, you mean like a large molecule? And would that be applied to, say, a brain tumor, a glioma? What would that mean?
So a polymer might be a plastic, or it might be a rubber. You know, those are examples of polymers, both plastics and rubbers.
Okay.
A molecule could be a drug, it could be a large molecule, or it could be a small molecule. It depends on the situation. And yes, you can apply it to many kinds of cancer. I mean, we've been involved in doing research that's led to new treatments for brain cancer, for advanced prostate cancer, and some other tumors. But they're not limited to that. You can really apply these principles to many diseases. Some companies have applied the principles we've developed, for example, to new treatments for schizophrenia, new treatments for type 2 diabetes, new treatments for alcoholism. So they're not, the principles are general. So once you have a deep understanding of how to manipulate the polymers or plastics, so to speak, and model the release, you can really apply them to almost anything.
Do you think that, are we on the verge, or is that a very difficult thing to imagine, the conquering of cancer, so-called?
Yeah, I don't know that I would go so far that we're on the verge of conquering cancer. I think we are, hopefully, in the next, say, 10 or 20 years, making significant progress. I mean, some clinicians, and I'm not a clinician, might say that the hope is to make cancer more like heart disease, that you
A
could...
maintenance situation like diabetes or...
Yes, exactly, and so that would be a hope. And I think that we are coming up with more, of an understanding of people's, not me personally, but I mean,
Sure.
a number of scientists are coming up with better ideas of people's genomes and how medicines might affect those, the individuals, and so you might come up with more individualized chemotherapy regimens for different patients, and I think that may help lead to the things that you and I just spoke about.
Let's stop for a second there. Individualized chemotherapy regimens. Now, this is a big center with Eric Topol of translational medicine. The fact that not everybody's body or genetics past is the same. Do you think that when you said targeted, will we be able to, using some of your methods, have a kind of individualized approach or therapy? Is that what you mean?
I think we will. I mean, both by what we're doing and by what others are doing. In other words, I think that, sometimes the individualized therapy may be just a regimen of drugs for different people. Sometimes it might involve targeting drugs to specific cells. And sometimes, like we're even creating, for example, little microchips that you can put different medicines in and get individualized release patterns.
And some of those release patterns are remarkable. Is it true that you can do that over a year, even 10 years, and therefore treat certain...
If the drugs are stable enough, or you can stabilize them, then you could do it over almost any length of time.
This is a funny question, does that raise any kind of security issues if somebody had an implant for 10 years? Or?
Well, I think it depends how the implant works. I don't think it... I mean, it's always possible that it could raise issues, but I think there's so many other issues that would come first, in terms of just getting the science to work. So I'm not so worried about security issues. And even with some of the chip things that we've developed, I mean, there's a lot of encryption techniques. You could put warning systems in, so I'm not that worried about them. And so many of them are for things that, I just can't imagine, like we're doing work to help people in the third world, in Africa and things like that. So I mean, I'm not sure why even a terrorist would want to, interfere with something like that.
Well, I just meant privacy, but
Privacy. I don't think I don't think that's going to be I don't think that'll be a big issue.
And you mentioned schizophrenia. Is there kind of an opening for these techniques for what is called mental illness or certain neurological issues?
Oh, absolutely. There are in fact, that's being done now. There are companies like Alkermes, which I was involved in for many years. I'm not involved in it now. But, I mean, they've done some wonderful work with little microspheres that you can inject for, say, up to a month to treat schizophrenia. I think millions of people have used them. There are systems that they've developed where you can give once-a-month injections for treating alcoholism, once-a-month injections for treating narcotic addiction. So... and I think that area in general is one where, better drugs are needed, and I think, better delivery systems are needed.
Well, that's incredible. And then the other thing that people talk about so much is tissue engineering. And I thought it was fascinating, almost a biomimicry approach. Explain that kind of layering of platforms, and then the seaweed concept for the three,
Sure. Well, tissue engineering, that's something I got involved starting with Jay Vacanti, who's a good friend of mine. Jay is head of pediatric surgery at Mass General. And I remember he must have come to see me in 1983, 32 years ago, because he would treat liver transplant patients. And the only way he could treat them is somebody else would die, and he would do a transplant.
Yeah.
And so we started talking about ways that we might be able to do better someday. And one of the thoughts we had is that we could take cells and put them on materials, and if we created the right materials with the right orientation, maybe the cells would reorganize and create a tissue-like structure or an organ-like structure.
It really is amazing.
Yeah, and that's happening. I mean, it certainly took some time, but now you can make artificial skin that way for burn victims and for patients with diabetic skin ulcers. You can, there are clinical trials ongoing now for making new cartilage, new bone, new cornea, even new spinal cords.
It's fantastic.
Yeah, no, it's wonderful. Yeah, and it's helping, potentially to help new blood vessels too, to help lots of people.
Well, the idea of was it that doctor who was vacationing in Cape Cod? And
Well, yes. So how we got the scaffolding, that's Jay Vacanti, that was my friend, and when we were trying to get enough, what should I say, surface area to volume ratio, one of the things he saw was seaweed when he was in Cape Cod, and he called me up and said, could I make a polymer that looked like the seaweed? So we did. And that did get us enough cells on the, enough cells per unit volume to make a difference.
What do you think will be the next revolution, or what's going to be really big both in health or biotech from your point of view? What do you see out there on the horizon? Maybe we can't attack it yet, but what do you see?
Well, I think there's a number of things that are going on, and I think it depends on whether you look at it from a technological standpoint or a disease standpoint. From a technological standpoint, I think there's now new kinds of drugs that are potentially, revolutionary drugs based on turning genes on and off or even editing them. So there's drugs like siRNA, which is a way to turn genes off, and those are now in advanced clinical trials, actually, treatments for that. Then there's mRNA, where you could modify messenger RNA and actually turn a gene on, or DNA to turn a gene on. And then there's even approaches where you can edit portions of a gene. All these are going to depend, I think, heavily, their success will depend heavily on delivery systems, delivering to the cells you want in a safe and effective way. So I think technologically that's one area. Another area, though, is just information. There's so much information that you could probably obtain from people if you had the right ways to do it, and you could maybe figure out patterns of by getting their information about how you might treat disease. And so I think having the right sensors that could collect information, that could analyze it, that could tell you how you might better treat different populations of patients or individualized patients is another big area. And then I think on the disease front, there's just so much we need to learn. It would be great if we understood more about cancer and how cells, change their nature to become abnormal. It'd be great if we could learn about Alzheimer's diseases or brain diseases and figure out, how to treat those brain diseases, and same thing with heart disease, but so many diseases. So I think that there's, a tremendous amount of both basic and applied research that, we'll see over the next 10, 20, 30 years.
And you're so well known for starting companies, and you have one of the largest labs, 100 scientists and postdocs, that sort of thing, back at MIT. But you have a very entrepreneurial view, it would appear, to how science can contribute to health. Could you speak to that? Why do you feel that way?
Yeah, well, what I found when I started, my postdoctoral career and then as a young professor, is like I would write papers, and those papers were in, good journals like Nature and Science and other ones, PNAS and so forth. But I'd write these papers, and, people would cite them and they might use them for some academic research. But I just thought naively that they'd also lead to new medical treatments, and
Right.
They didn't right away. And then what happened was, about 10 years after I started, there were several large companies that called me up and said, well, could I consult for them? And I did, and then I mentioned even we had some of these patents that we had applied for, and that they could license them and maybe develop them, and they did actually. They licensed them, they gave me grants and things like that, and I was very excited. I thought, gee, our work is going to get out to the world. But those large companies didn't do very much. I don't think
Right.
they had the same passion that, say, the people in our lab did, and they were working on a lot of different things. So I was able to get those patents back, and then we use those patents to start companies, because my friends would tell me, that it would be exciting to do that and important, and I was very interested in seeing our work get out to the world, and I could see that these small companies were you know, the passion was there. The people working at the companies were often people who'd worked in our lab, who wanted to see their discoveries get out to the world. And that worked. I mean, though that's what's led to all these treatments. It really moved from our lab to these companies, and it's the students and postdocs and the other people at the companies that have just done a wonderful job that have, you know, created these products. And I think that that's been what's been so great about so much of America, that you see these companies starting up. You know, they start, you know, sometimes with one or two people, but sometimes they grow and become bigger and bigger, and, you know, create products that can just help so many people, and I do believe in that.
Absolutely. I mean, San Diego is one of the pillars of the economy here, is biotech.
Terrific,
And a number of companies, and up in LA we have Amgen,
Exactly.
and Illumina here, and HR, and any number of other companies.
That's right, and they
you're
all start.
involved- Go ahead.
Yeah, and they all started tiny. Yeah.
Which companies have you started, or do you feel involved with these days?
Yeah, well, I mean, Alchemies, which I mentioned, was based on some of our work. I've been involved with Alnylam from the beginning. Moderna is a newer startup that's doing some of these RNA things. Momenta is another one that we started that's doing a lot of work in the polysaccharide area. But there have been quite a few. I mean, I've been on the boards of many different companies, and then there's newer startups that we've created in different areas doing nanotechnology, like, you know, Bind and Selecta and Blend. I mean, there's a lot.
No,
I
but
mean,
it's interesting to know. I mean, our audience is,
Yeah.
there's tens of thousands, if not 100,000 people in the San Diego area that
Sure.
work for biotech.
Yeah, but I mean, I think all of it's great. I mean, you know, some will do better than others, but I mean, all these places are doing things that, you know, can really improve people's lives, and it's to me, it's like a win-win-win. You know, you're creating jobs, you're creating technology, and you can create products that'll help people someday.
That's awesome, as we say out here.
Yeah.
And you know, I was in Cambridge in the early 70s as well. We talked about that before the interview. I mean, there was, they weren't called disadvantaged areas. There was a white slum, the projects. We lived on River Street, Pine Street.
Right.
Between Harvard and MIT, it was a pretty rough, you know, neighborhood.
Yeah.
And then you, for some reason, I'd like to hear why, became the head science guy at a famous place then, the group school. You seem to feel that kids from disadvantaged backgrounds or had bad attitudes could learn science and math. How did you do that? Why, and how was it successful?
Yeah, well, when I was an undergraduate at Cornell, I did tutoring, teaching system, things like that. And when I came to MIT, I did some tutoring in Cambridge. And the year after I came, I guess I was doing okay and doing some of it. And there were some people wanting to start a school for disadvantaged kids, poor kids, and they asked if I would help, and, you know, and maybe help, particularly on math and science. And that was a tremendously exciting experience. I mean, I remember when I came that year, and you know, they had like 37 students, and of course this was Cambridge in the 70s, so their attitude about science and math was, it wasn't required. You could take it if you wanted to, but you didn't have to take it. And so I felt like I wanted people to take it, not because they had to, but because they wanted to, and so I wanted to figure out ways to make it interesting. So I remember the year I came, 5 out of 37 people took it. By the next year when I was doing it, 45 out of 50 took it. And same thing with chemistry. We developed, I got MIT to fund a program where myself and two other people, David Sincata and Kathy Cole, we developed a new chemistry curriculum to try to make chemistry exciting. And
Wow.
that was also very popular. And so, and then I also created these programs at MIT where people could get summer jobs from the school, and I think that was, that also ended up being very, very exciting. In fact, one of my interesting story, one of the students I remember that was at that school was a guy named Joey Boyle, and it must have been about 25, 30 years later. I was, Chuck Vest, who was president of MIT then, asked me to give a lecture to the incoming class at MIT, and I did, and at the end of it, the student comes up to me, who was a freshman at MIT, who was actually his son, and he said you know, I mean, and he was, you know, going to MIT, and it was just wonderful to see, you know, how a school like that could affect people's lives.
Is it possible to say in a few words what does excite younger people about science and math? I mean, kind of hard cases?
Well, I think relevance does. You know, I think showing you that you can take science and math do something important, I mean, not unlike what we try to do on research. In other words, I think it needs to be more than just abstract. It needs to, I think when you show people that science and math can do something to make something good, to make something useful, I think people get more excited about it. I certainly do. That made a difference to me my whole life, and I think it does for lots of people. So that's always been the guiding force for me. Like I remember teaching thermodynamics for several years as a chemical engineer, and that was challenging to try to get students excited about it. But when I could try to show some relevant examples of it, that you could, make new drugs that way or new drug delivery systems that way, I think students got a lot more excited about it.
Cool. And is there a retreat from science? I mean, the cover of National Geographic this month is the war against science. And on the other hand, NIH funding, National Institute of Health's funding is an issue now. Francis Collins was out at Scripps last week. Do you think that is going on, or at the same time there are all these great discoveries and wonderful things going on at MIT or UCSD?
Well, I'm concerned about funding for basic science and basic engineering. I think that's a big issue, because that is harder to get, and I think it's particularly harder to get for young people. And to me, those discoveries are gonna, that's the future. So it's not to say that people aren't making them now, but I mean, I want 10, 20, 30 years from now things to be as good, if not better than they are now. And if you have less funding for basic science and engineering, they won't be.
Okay, and congratulations on being a Kyoto laureate.
Well, thank
Kyoto
you.
Prize is a wonderful thing.
Thank you.
So what's next for you? I mean, you're a young guy.
I don't know if I'm that young, but I love what I'm doing. I mean, I guess I feel, we're gonna keep doing research that I hope can, push the frontiers of bioengineering further, and training students who are, leaders in bioengineering. I have a number of people from my group who are professors at UCSD and many other places. I think there's something like 270 of my students are professors now, but a number of them are out here. And I mean, those things, and we're going to keep trying to turn our discoveries into companies and products that I hope will improve people's lives.
Okay, fantastic.
Thank you. We're going to try at least.